Hello, my name is Brett Arenz. I am the director of the Plant Disease Clinic in the Department of Plant Pathology at the University of Minnesota. The Plant Disease Clinic is a service lab that diagnoses plant disease from samples submitted by government agencies, farmers, professional horticulturalists, as well as the general public. So plant health problems can be very frustrating if you grow plants, especially if you depend on plants for your livelihood. And diagnosis of plant health problems is really the essential first step to managing those problems. However, to get an accurate diagnosis, you really need to start with a high-quality sample. So what we have here are a couple different plant samples which were submitted to the Plant Disease Clinic. We have a juniper here and a hosta, and these are high-quality samples, not in the sense that they're healthy, because they're obviously not, but in the sense that they have a lot of tissues that we can work with when we make our diagnostics. They contain both living and dead tissues. It can be actually very difficult to diagnose a sample that is completely dead, because at that point other microorganisms have begun to colonize those dead tissues, and it can be difficult to actually find the microorganism that caused the disease in the first place. So what we're really looking for in a lot of cases is that area between the green and the brown tissue, so that area between healthy and dead plant tissue. Those are areas where the pathogens are most active and where we can more reliably find them. Another reason these are good samples is that these are whole plant samples, so they contain both above ground and below ground tissue. In many cases, pathogens that are actually affecting the roots will cause symptoms in the leaves. However, if a client just brings us leaves, in many cases those pathogens aren't going to be present on those tissues, and so we can't make an accurate diagnosis. So it's always good, if possible, to bring it in a whole plant. And so of course these are two potted plants. These were brought in by hand to the clinic. That might not be possible for you. You might live too far away from the clinic, and so you want to mail in a sample. And so what I'd like to show you next is actually how you can mail in a whole plant sample and how you can package them in a way so that when they arrive at the clinic, we actually do have a high quality sample to work with. Okay, so what I've done is I've taken the hosta out of the pot and I've shaken some of the excess soil off. And so I have put a plastic bag around where the roots are, and I have just tied that off with a little twisty tie to keep it nice and tight. The reason you don't want to put the whole plant sample in a plastic bag is you can get a lot of humidity that's going to collect in there, and then the sample is going to degrade pretty rapidly if it's in the post office for any length of time. But if you can wrap the roots off, that should prevent it from drying too much. And so to the above ground part of the plant, what we can do is just simply take some newspaper and wrap some of those leaves up. And you want to make sure it's dry newspaper. If it's wet newspaper, that's just going to contribute to the sample degrading too much during shipping. And so then we're going to carefully put it in a box, and the newspaper really is just there to prevent the sample from moving around too much. So even though we package the samples correctly, we still want to mail the samples so that they arrive in the clinic in as little time as possible. Try to avoid mailing samples on a Thursday or Friday if it means that they're going to sit in the post office over the weekend. You can find more information on our website at pdc.umn.edu. You can always give us a call as well at 612-625-1275 if you have a question about this.